Okay, just to make sure that I'm not encroaching on the coffee break too much, I will just jump right in with my talk. And I'm going to talk today a little bit about my PhD project, which had the title How Acute Stress Modulates Hemispheric Asymmetries, Investigating the Role of Endocrinological and Affective Parameters. So, one second. We have heard a lot about hemispheric asymmetries in this conference, and one of the structures that has been implicated in the maintenance and establishment of hemispheric asymmetries, especially functional hemispheric asymmetries, is the corpus callosum, namely the connection between the two hemispheres. And it has been proposed that it does so by inhibiting the non-dominant hemispheres. So each hemisphere is dominant for a certain task, and activation in the dominant hemisphere inhibits the non-dominant one. The corpus callosum does so in that it is itself glutamatergic in transmission, so excitatory, but it synapses on GABAergic interneurons in the contralateral hemisphere, resulting or probably resulting in a net inhibitory effect. And there are several factors that can actually influence functional hemispheric asymmetries. So, for example, multiple genetic loci have been implicated, as well as certain intrauterine effects. But last but not least, also environmental influences can affect functional hemispheric asymmetries. So, for example, lateralized stimulation has been shown to have a great effect, especially in pigeons and chicks. Also training effects, for example, if you think about sports, there is a strong effect on training, but also hormones have been shown to affect functional hemispheric asymmetries, especially through the influence of the corpus callosum. And here, more so, sex steroid hormones have been implicated. For example, progesterone has been shown to downregulate glutamatergic activity and upregulate GABA, which causes a decrease in inhibition through the corpus callosum, which actually means that there is an increase in bilateral activation, which is basically synonymous with a decrease in functional hemispheric asymmetries. Estradiol, on the other hand, has been shown to upregulate glutamatergic activity and not really have a big effect on GABA at all. So this increases the activation in both hemispheres, and depending on which hemisphere is dominant, it changes functional hemispheric asymmetries. So following this logic, we were interested in if other hormones also can affect the transmission of the corpus callosum, and especially the functional hemispheric asymmetries that come with it. And here we were interested in stress hormones. There are two different systems of the major stress response in humans and other mammals. And the first one is the sympathetic nervous system. It starts with activation of brainstem nuclei, which causes secretion of epinephrine and norepinephrine from the adrenal medulla and the postganglionergic spinal cord neurons. And the other one that I've been focusing on mostly is the hypothalamus pituitary adrenocortical axis. And as the name already states, it starts with activation of the hypothalamus, which secretes ACTH, which causes the anterior pituitary to release CRH into general circulation. And that causes the adrenal cortex to produce and release cortisol. And cortisol in turn can act on all cells of the body and also cross the blood-brain barrier and affect the brain. So we set out to basically test two different models that state that there might be an effect of stress or acute stress on hemispheric asymmetries. And first, we wanted to test the affective model. So we know that emotions are processed laterally, and negative emotions are most predominantly processed in the right hemisphere. And stress actually causes an increase in negative effects. So we could guess that this leads to a selective priming of the right hemisphere. So we would expect that functions that rely stronger on the right hemisphere profit more from a stress effect. On the other hand, we suspected that in the psychoneuroendocrine model, the effect of acute stress might be more related through the corpus callosum, in a way that stress can actually, and especially stress hormones and cortisol, can affect glutamatergic and GABAergic transmission. And here we would expect an increase in inhibition and thus hemispheric asymmetries. So for asymmetries or tasks relying on either hemisphere, we would expect an increase in asymmetry. So these were basically the studies that we performed for this project. We had two studies with behavior, ones with stress, ones with hydrocortisone to actually decipher if it's actually a stress effect or if it's a cortisol effect. And then we performed 2 studies with EEG, again, ones with stress and ones with cortisone. Coming to the experimental design of the very first study, we had participants come for 2 days. On one day in the lab, they performed a stress induction paradigm, the triasocial stress test, a very established paradigm to actually induce strong feelings of stress in our participants. And afterwards, participants completed 3 different tasks that I'm going to into detail a little bit in a little bit of time. And we also had participants give saliva samples for measures of stress hormones, as well as questionnaires about their personal feelings of stress. And in a second day, they underwent a control procedure, which is very similar, but that doesn't cause stress in the participants, just to make sure that everything was counterbalanced. The timing, the order was randomized across participants, and we tested 60 participants for the first study. So for the tasks, first we had participants perform a verbal dichotic listening task, which measures language lateralization. I don't think I have to tell you a lot about it because you all know this task. 2 syllables, one to the left, one to the right ear. Participants have to indicate what syllable they hear best. Typically, you expect a right ear advantage. We also had participants perform a emotional dichotic listening task, which should measure emotional lateralization. So here participants got the same syllable pair to the left and the right, but on each side, it was spoken in a different emotional intonation. So here participants had to indicate the emotion that they perceived best. And here we would expect a left ear advantage indicative of right hemispheric dominance. For all of these measures, we calculated a laterality quotient, and you will be very happy to see that greater positive values will be indicative of right lateralization, and greater negative values will be indicative of more left lateralization. Participants lastly also performed a Barnich-Berger task, which is used to measure interhemispheric integration. And here participants are presented with a fixation cross in the middle of the screen and 2 letters above and 1 below the fixation cross. And now they have to indicate via button press if the lower letter coincides with either one of the upper letters or not. And there's different conditions in which this can take place. First, the physical matching condition, in which all letters are uppercase letters, which is more easy because they only have to match the physical appearance of the letters with each other. The name matching condition, on the other hand, is a little bit more complicated. Because the lower letter is lowercase, so they actually have to match the identity of the letters. And usually here we would expect that participants perform better on the cross-field tries, as they profit from integrating the information across 2 hemispheres. And this is called the cross-field advantage. To directly go to the results, here you can see the lateralization quotient for correct responses, and here for reaction times. Blue is the stress condition, yellow is the control condition. For the verbal dichotic listening task, we found a typical right ear advantage. For the emotional dichotic listening task, we found a left ear advantage. And what you can see in the difference between the 2 conditions is that there was no difference. So we still kept on looking a little bit, I can also spoiler the across field advantages and all the indices in the Barnich-Baker task also were not different, but we didn't give up yet. So we calculated some correlations between different indices of stress and the across field advantages. And here we found in the stress condition that there was a positive association between salivary alpha amylase, which is an indicator of sympathetic nervous system activity, and the across field advantage. So this suggests that if they had higher acute stress, there was better interhemispheric integration. To the second study, we basically did the exact same thing, but this time participants were not stressed. They were giving 20 milligrams of hydrocortisone as well as a placebo in 2 test sessions again. And we also had the 60 participants with funnily exactly the same division in male and female participants. So directly to the results. Again, here the LQs for correct responses and reaction times. Blue in this case is hydrocortisone. And again, no differences. Absolutely nothing. And this time also we performed the same correlation analysis. And also there were no associations between any LQs and cortisol scores or salivary alpha amylase scores. Also, we confirmed our results with Bayesian statistics, and there was no association between stress or cortisol and any lateralization measures. Finally, we performed the first study with EEG. Participants were equipped with the EEG cap. Then they underwent stress induction or control procedure, after which they performed a resting state EEG just to get into the groove of the whole EEG measurement thing, after which they performed 2 different tasks. Here, the first task was a Poffenberger paradigm, which measures basically transmission properties of the corpus callosum. Participants are presented with a fixation cross in the middle of the screen and a bright dot either to the left and the right. And they are instructed to press a button as soon as they can see the dot, which actually does not really interest us at all. We are looking at the EEG signal at the O1-O2 electrode pair over the occipital cortices. And the idea is that if the stimulus is presented in the right visual field, it first enters into the left cortex and from there has to be sent to the contralateral side. So we would expect a latency difference between the 2 electrodes dependent if the stimulus is presented ipsilateral or contralateral. And this is called the interhemispheric transfer time. So the time it takes for the stimulus from one side to be sent to the other. We also had a lexical decision task, which should hopefully measure interhemispheric communication for language stimuli. Very similar to what we've just heard, participants had to indicate if they were seeing a word or a non-word. And very akin to the Poffenberger paradigm, we would expect that it would take more time for one N1 component compared to the other, depending on ipsilateral or contralateral presentation. And here we took this as an indicator for interhemispheric integration. So to the results. This is the N1 latencies for the Poffenberger paradigm. And here you have the different electrodes with the different visual fields in presentation. And to make it real quick, you see that we are faster for the contralateral presentation. So at the O2 electrode pair, it's faster compared to the O1 electrode pair. If it is presented in the left visual field, which is the exact Poffenberger effect that we hoped for. Also, again, to the stress effect, no effect. Surprise. For the lexical decision task, we basically here have the N1 component as well. And here we found a small but significant effect of shorter latencies if stimuli were presented in the left visual field in the right hemisphere after stress induction. So there seems to have been a faster information transfer from the right to the left hemisphere under stress. So to corroborate our results, we repeated the study with, again, hydrocortisone compared to placebo. And to make it very short, no main effect in the Poffenberger paradigm, also no main effect in the lexical decision task. So just to summarize real quick, what did we find in all of these studies? What did I actually find out in my PhD? Well, on a behavioral level, stress does not really seem to affect hemispheric asymmetries. We found a small but significant effect, positive effect on interhemispheric integration. So interhemispheric integration across both hemispheres seems to profit a little bit from stress and the appearance of stress hormones. Second, hydrocortisone did not result in the same effect as stress. So neither on hemispheric asymmetries nor on interhemispheric interaction. Third, we found improved interhemispheric interactions in a lexical decision task after stress induction, which was not there again if we did the same thing with hydrocortisone. So what does this mean for the two models that I presented to you in the beginning? If the affective model would have been true, we would have expected higher asymmetries in tasks that depend on the right hemisphere, which would have been the emotional lateralization task. We would also have found a association probably with negative effect that we measured after stress induction, and we found neither of those. If the neuroendocrine model was true, we would expect higher asymmetries for all tasks, also in association with the cortisol level, also in the hydrocortisone studies. And we also did not find this. So this leads me to conclude that neither of these models are supported. This is actually a little bit at odds with other literature or older literature that found some effects of acute stress on hemispheric asymmetries, but we could not replicate any of these results. This actually leads me to conclude, at least a little bit, that there might just not be an effect of acute stress on hemispheric asymmetries whatsoever. And yeah, with this, I would like to thank you for your attention and thank my collaborators, and I'm looking forward to your questions.
Thank you very much.
Thank you. That was really interesting. I wonder if you could take me through the timings from the moment after the stressor ended and where that sat amongst the tasks that were completed.
Yes.
Because as we know, cortisol peaks only about 20 minutes after the end of the stressor.
Exactly. Can I wait a second? I will just go to the slide. Yeah. One moment. Can I move this one? I will just do it like this. So yeah, for example, I go to the one with the stressors. Bless you. So the stress induction itself takes about 15 minutes. You have a 5-minute pre-stress preparation period that also counts as a stress induction period already. And then you have 5 minutes of an impromptu presentation. It's basically a mock job interview, and then 5 minutes of mental arithmetics in front of a very judgy panel. And after that, the salivary samples as well as the questionnaire take about a minute tops. And each task was about 10 minutes time. So we have like from the beginning of the stressor, then you have the first task, it's plus 15, plus 30, plus 45. In case of the placebo study, this was very similar. Obviously, we had to wait a little bit until the placebo could dissolve in the stomach and then be absorbed. So this time period is 40 minutes because that's just how long it takes. But then it's again, I think, 12 minutes, 12 minutes, 12 minutes. So it was very comparable. With the EEG, no, this was also 10, 15, and 10. This is both 12 minutes. So these two are both 12 minutes. This is about 5 minutes, but here participants took a little bit longer because it's a little bit more complicated to navigate with the EEG cap on to make sure that nothing gets disrupted.
Okay, thank you. And if I can ask just one more question, I was wondering at an individual level, did you look at whether you had differences in cortisol responsivity? Maybe you had cortisol responders versus non-responders, and if that was accounted for?
Yes, we looked at obviously we looked at the cortisol. I didn't present the data just because of time constraints. We actually had very good cortisol reactions. We had a student assistant who was always on the panel, who was seemingly very good at judging our participants and inducing a stress response. We never had these great cortisol reactions before or after. But yeah, we had a few participants that showed a very minuscule cortisol reaction, but taking them out didn't change the data either way.
Just following up on that from my side, does time of day influence the data?
Yes, it does. But that is the reason why we only tested in the afternoon, because cortisol has a very typical peak in the morning, and then the secretion slows down across the day. So to make everything comparable, we only tested in the afternoon.
I have another question.
Oh, yes.
Is it an accident that you only tested men in experiment 3 and 4?
For the EEG analysis, it was a little bit a combination of convenience and theory-driven design, because for the women, it is a little bit more complicated to find participants that are not on any hormonal contraceptives. And that made it very difficult for us to actually make sure that they were on the same cycle period, and especially because the EEG, at least I think so, might pick up on more differences in asymmetry than a behavioral measure. I wanted to make sure that the participants were comparable between the two test sessions. That's why we decided to exclude the female participants.
Yeah, that's exactly what I was going for, because I've heard that there's a difference in cortisol response when
Yes.
you're on the pill. Or
Exactly. There's a difference in cortisol response if you're on the pill, and there's a difference in cortisol response and in asymmetries across the female menstruation cycle. So to make it comparable, we opted to exclude the women. Obviously, it would be better to have also the same amount of women, but it was a PhD and I didn't have unlimited time.
Other question?
Yeah, nice talk. Thank you very much. I have a question regarding the responder and non-responder. So did you maybe find cortisol effects on asymmetries? So were there, in terms of the asymmetry stress responder? So obviously, I mean, you managed to induce stress and people responded to that. I think that's great. But the question is, did the stress in some participants have the effect that you predicted? And maybe not in others for whatever reasons?
Yeah, we looked at this in the very beginning. And we basically did an analysis on extreme groups where people who showed a very pronounced stress response actually showed a little bit of an effect. But I would be very careful with interpreting this data, because obviously it's in this extreme group, and that actually makes it like a very small sample size. I think it was like 13 participants in the end. So I would be careful with interpreting the data in that very small sample. If there was the possibility to maybe repeat the whole study with an even larger sample size, this might be a great way of analyzing the data. Yet still, we didn't find anything in the correlation analysis with cortisol, so maybe we would have seen something anyways. I don't know.
Okay, thank you very much again.